Okay. So the concept here is beyond. At how many levels can you study nutrition in terms of the effects on a biological system, but also maybe even at an ecosystem level? How does that fit? And how can we measure all these effects that might happen through what we put in our mouth every day of our life? So this becomes rather amazing in this era that we live in, because we can measure such impacts at many, many, many levels. And this is what I'll be going through. The effect of nutrition in terms of health, of course, was captured very well by Hippocrates many years ago when he said, let food be your medicine. And in a lecture like this that I gave some years ago, I tried to encapsulate the concept maybe of systems health and systems medicine, perhaps, rather, and nutrition, by trying to write a version, let's say, similar to the Hippocratic Code that encapsulates these new sciences and these new ideas. And I'll finish at the end with that. This is only a draft, and people have many different opinions about such a way of presenting the concept. But rather than just putting a list of take-home messages, I thought maybe these ideas can be reworded in this concept that Hippocrates put to us, and simply see whether it makes sense in the framework that he has recommended to us. So the points that I'll be covering here are the principles of genome, epigenome, transcriptome, all the omes, so the multi-omic approach, some of the biomarkers that are used to determine nutritional status, and some ways of representing this omic data through network maps and so on. These days, nutrition, we have to think of nutrition as part of the whole exposome, or all the exposures that we experience in life. The exposome concept was first presented by Chris Wilde, who was then the director of the International Agency for Research on Cancer. He did a lot of work on aflatoxins through studies in Africa and their impact on cancer, for example, which comes through food. But of course, there are many environmental factors. It could be psychological stress, lifestyle, infections, and so on. And diet is part of this. And we have to therefore consider diet in the context of all the other exposures also that we experience. I showed a slide yesterday. It's just a slide there to remind us that we come from many different countries, from different cultures and backgrounds, all of these impacting in some way also evolutionarily on our genetic diversity, and also the epigenetic aspects of this genetic diversity as well. And the variation is both in the amount and the quality, and the variation of and types of food, and how that interacts with our genome, and is obviously the topic here, and how we measure those effects. So there's great diversity in nutriomes and genomes. And before we even start to think about nutriomes and the genomic effects, we really need to start also thinking about the foods themselves and how they are interrelated to each other in the foods that we eat. A year ago, I came across, I had already been, well, we are all thinking about, how can we feed the world, and what's the minimum set of foods that we might need to feed the world. In other words, which are the nutrient-dense foods, and how can we put them together to meet our needs? One way you can do this is by mathematical modeling and bioinformatics and statistical methods, as has been done by Song-Gyun Kim in Korea, just unpublished this work a year ago. He's actually not a biologist, or his group are not biologists and nutritionists, but really he is a physicist, if I recall correctly, with great skills in data analysis. And what they put together here is a network analysis of the nutrients in foods and how they tend to be related with each other. So when you see a blue line, that is a positive correlation. If you see pink line here, it's a negative correlation. And what you can see are some relationships that are quite interesting. So if you're eating protein, you're likely going to be rich, have foods rich in selenium. Some foods tend to, nutrients tend to occur together in foods, such as vitamin A and vitamin K, for example. And you can find all kinds of other relationships like this that help you to define how foods could be put together in a way that you can achieve the desired nutrients, depending on the health outcomes that you want to achieve. Furthermore, this modeling can be used to determine the minimum food sets to meet the requirements and nutritional fitness of foods. So what the nutritional fitness of a food as determined here was based on the food's frequency of occurrence in nutritionally adequate food combinations. So the mathematical model throws up all possible food combinations that would meet the minimum needs. And the food's nutritional fitness depended on how frequently it occurred within those sets. And this was the system that they used to derive these minimum food sets. And what you can see here, the very top of the nutritional fitness field would be things like almonds, ocean perch, chard, and so on. Whilst further down you'd find foods such as beef, pork ribs, sesame, and so on. So this depends on the model, of course, that you are using and how you interpret that data. I'm sure this is only a first iteration of this possibility of looking at foods, but it starts to give you a hint of how foods might be put together in more than one way, depending on the context and the country where one is living. Now, another way to look at food is to say, well, is to try to understand rather which cofactors we get from food that interact with proteins and that might affect, therefore, metabolism, and which of those proteins that require cofactors from food are actually linked with disease based on genome-wide association studies that identify the genes and therefore the proteins. And in this paper published just this year by Scott Boyer and others in Scientific Reports, they do a network analysis of the cofactor protein interactions for analyzing associations between food and disease. And essentially what they're looking for is the cofactors that interact with proteins and which of those proteins actually are found to be associated with disease. And on this basis, they developed a network of such information. And the network is shown over here. So you can see here for each cofactor, be it a mineral or a vitamin, you can see it's associated with a number of proteins. So let's take zinc here on the left. You might not be able to read it. It's associated with many, many proteins. And some of these proteins might be associated also with another mineral or another metabolite, in this case, acetylmethionine, which is a common methyl donor that emerges, that is synthesized when you eat foods that are methyl donors, such as methionine, folate, choline, and so on. So in this way, you can get an appreciation of the interconnectedness of essential nutrients, micronutrients, and proteins, as well as with proteins that are important in disease if they don't function well. Because the fact is, it's not just mutations. You might have, let's say, the Olympic gold standard versions of all the genes that a human could possibly need. But if you don't have the cofactor that protein that gene codes for needs to work and function well, then it's as if you have a mutation in that gene, in the sense that it will not work as well as it ought to. On the other hand, you might have a mutation that could be overcome if the intake of that nutrient was over and above what is normally needed in a person who's got the optimal version. And an example of that would be the methylene tetrahydrofolate reductase mutation that requires more folate or more riboflavin to correct the phenotype. Now, this network with proteins then can also be connected to the diseases where those proteins play an important role. And essentially, using this information, you can then start to devise or think of ways how a disease might be attenuated or cured or managed better by optimizing those nutrients needed to make that malfunctioning protein work better, or at least to find another, to support another metabolic route that might overcome the metabolic block that's causing the disease. Another way of understanding the exposome and the role of nutrients is to use the same tools for genome-wide association studies, which are statistical methods to make an association between mutations in genes and disease. But instead of using genetic information to produce these so-called Manhattan plots, which tell you about the linkage of the factor with a disease condition, in this case type 2 diabetes, you put in your model the environmental factors that the persons are exposed to. Now in this case, in the study by Patel et al. In Stanford, they made this association between exposures to environmental factors such as nutrients, such as micronutrients in the blood, pesticides, and other factors in the environment. And what they found was that type 2 diabetes is associated, showing protective effects of beta-carotene, a aggravating effect of gamma-tocopherol, which is also associated inversely with the health eating index, which I'll show later, and exposure to pesticides. So this is another way of looking at risk factors. And given that environmental factors are thought that in many cases to have a stronger effect than genetic factors, it's very important to also look at the environmental issues and nutrition in this way, and ultimately to integrate this with the GWAS, or genome-wide association study data. Now, now let's get into the omics, which is really the system things that we think about. But I thought before we got into these omic markers and factors, we really should understand the nutrition part very well. So that's why I started with this aspect. So on the omic side, we can start, of course, with the we've already started and talked about the nutriome, which is the nutritional combinations of the diet or the foods that you prefer to eat or choose to eat. But the nutriome has an impact, as we know, on the genome to begin with, on genome stability and the epigenome. And you can measure this at a molecular level by looking at the ratio of, let's say, cytosine and 5-methyl-cytosine and this kind of thing. Or you can look at breaks in the DNA, or you can look at DNA damage at the chromosomal level, at the cellular level, at the cytogenetic level. You can look at the impact on the transcriptome, on which genes are being expressed, on the microRNAome, which is another level of regulation of gene expression, on the proteome, the metabolome, and how this impacts on health and well-being. So let's start with the genome. So the genome, of course, provides the genetic information. This genetic information in DNA is packaged in a particular way on histones, and the histones themselves can have impacts on the expression of the genes. Within the DNA, there are sections or sequences at which transcription factors can bind and activate the expression of a gene. And there's also the transcription machinery that enables the transcription of the genetic information to make RNA. And as I mentioned yesterday, a large proportion of the DNA is not protein coding, but RNA coding, which actually then further regulates the expression of the genes. Now, our knowledge about the human genome started some years ago with the Human Genome Project, where the sequencing of the genome of Homo sapiens was performed together with that of some of our primate relatives. Ongoing is the 1000 Genomes Project, which is identifying the variations between individuals across the whole of the world. Concomitantly, there is the Cancer Genome and Epigenome projects going on. So there are also what are called, for example, the epigenome-wide association study, not to be confused with the environmental-wide association study which we saw previously, to characterize differences between tissues, let's say, and between tissues when they are healthy and when they are diseased, and then to utilize and decipher this information to understand cellular mechanisms. So we will have massive amounts of information, and this is growing rapidly in many ways. So coming back to genome-wide association studies. So what really happens here is that one is trying to associate in an epidemiological sense the mutations in the genome, either usually as single nucleotide polymorphisms or other markers within the genome, with disease. And that is done by comparing populations that are healthy and ones that are diseased. And then trying to find out via these Manhattan plots those regions of the genome that associate most strongly with the condition. And this is usually done in more than one study. Maybe at least 2 replication studies are required before consensus is achieved. Although this level of consensus will depend to a large degree whether the study was done in different countries or within the same country or population. Now, interestingly, you can, because there are so many GWAS studies done, you can also do what is called a meta-analysis. And that is further analyzing all the GWAS studies that have been done, where an association has been made between polymorphisms and age-associated diseases. So these are diseases that increase in frequency as we get older, such as cardiovascular disease, cancer, and dementia, let's say. And very interestingly in this study, when they did the meta-analysis, 2 hotspots turn up, which are enriched for genes linked to inflammation or senescence. And if you look more closely at one of these, for example, the 9p21 locus, there are a lot of genes very close to each other with hotspots for these diseases, such as Alzheimer's disease, cancer, atherosclerotic vascular disease, and type 2 diabetes. So there are some regions which are particularly important to understand how nutrition might affect mutations in this particular region. And this, I think, would help facilitate targeted approaches to understanding the impact of nutrition when it's deficient or in excess on disease, because it might provide some hints how we might intervene and prioritize what is more important or less important in the approaches we take. The field of nutritional genomics, which is what we're talking about here, is the impact of nutrition on the genome, really accelerated when gene expression arrays came on the scene as a technology. And in fact, this paper I'm showing you here is published quite a while back by Michael Muller, who was one of the pioneers in this field. He now heads the Nutrition Institute in Norwich in the UK. And he wrote a very nice review in Nature Reviews on this approach of gene expression arrays. And this is his diagram here showing this is the normal gene expression, so this is indicating health, and this is how the gene expression pattern might drift in the event of either poor nutrition or poor nutrition in combination with the associated metabolic stresses and inflammation. And thus one can identify stress signatures or the extent to which a particular individual is moving away from health, and the extent to which you might be bringing them back by bringing them back to normal. Now, the expression of genes, of course, is affected by nutrition, in many ways, and one of the most obvious ways is that some nutrients act as transcription factors. And for example, some fatty acids and cholesterol will act on transcription factors such as PPARs and FXR. Certain vitamins like vitamin A, which acts in this manner through the RAR and RXR transcription factors, and so on. In the case of zinc, it's an MTF1. However, this can happen either directly, so the nutrients may be direct ligands of transcription factors. Alternatively, they might modulate transcription factors, or they can act as activators of signal transduction cascades. So they might also be needed perhaps as cofactors within those cascades. Another way that genetics can therefore impact on the way nutrition controls or modifies gene expression is by mutations that we might inherit that are driven, that affect the ability of the factor, the transcription factor, to bind on the relevant sequence. So if there's a mutation here, in this case, a mutation of A to G, that might disable the ability of the transcription factor to bind and therefore not exert its effect. However, it may also be that that mutation has occurred not because you inherited it, but because you acquired it. And I'll show you a very interesting example about this. This is a paper published in 2004 from Yankner's lab. The first author is Liu. And what they did was they took, well, there were biobanked brains of people who passed away but who were healthy. And, well, they passed away because of an accident, and they donated their brains. And I looked at the gene sequence, the promoter sequences and gene expression in the prefrontal cortex. And what they observed was that oxidation of guanine in the promoter region of genes affected expression of genes which were changing with age. So there was, what they noticed was that there were some promoters were susceptible to oxidation, more so than others. And what they did was they studied oxidative damage in these promoters, and they showed that these genes involved in synaptic plasticity, such as the serotonin receptor, vesicular transport, such as tau and MAP2 and dynin, calcium homeostasis, such as calmodulin, and mitochondrial function, such as ATP synthase, cytochrome C, their reduced expression was occurring because of oxidative damage to the promoter. So this could be an indirect effect of nutrition. In other words, if you are not, let's say, eating enough antioxidants in your diet that may in some way impact the oxidation level in the brain, it could be this oxidative stress, which of course could also occur due to inflammation, may be impacting gene expression in this way. And here's in fact the gene expression pattern that occurs with aging. What you can see here is the gene expression pattern in the prefrontal cortex in those aged 20 to 42. This is in those aged 71 to 100, very different, because these are the genes that actually changed. And here in the middle age, 43 to 70, you can see it's intermediate. Okay? So it also instructs us that we need to intervene early, not late, to prevent this drift towards aging, to an inappropriate expression of genes with aging. You might be wondering how they measured oxidation in the promoter. It's actually relatively simple to do by PCR. What you need to do is amplify the DNA in the region of the promoter, but what you do is you treat the DNA with an enzyme called FPG. This is a glycosylase, which basically identifies the oxidized base. There may be other base lesions because FPG is not so specific to oxidation, but you could use HOGG1 instead. And then what it does, it nicks the DNA at that point. So then the PCR reaction slows down, and the delta of the CT, which is shown here, tells you how much oxidative damage happened. And this is what they showed, and you can go deeper into this paper. It shows, in fact, even the increase in DNA damage with aging for each of these genes which were downregulated. Okay, so let's hop now on to methylation, because this is the one that we think of most when we talk about gene expression. And I'll not talk a lot about this today, because tomorrow I'll be saying a lot more about this topic. But I'll simply show you something relatively simple to understand. So in the normal individual, in the normal situation, there are two aspects of methylation, right? So in the normal situation, this is an example taken from cancer, but it could be other conditions. In the normal situation, the tumor suppressor genes, which are really housekeeping genes, are meant to be active, in other words, meant to be expressed to maintain the normal homeostasis of the cell. So this is the normal situation. So you'd expect methyl-cytosine to be unmethylated, and you also expect the histone acetylated histone to be in this condition. And then there are regions of the genes which need to be suppressed, and these are genes such as transposons and repetitive elements. Some of this DNA are viruses, retroviruses that have been incorporated into the genome and really need to be suppressed. However, with aging, what tends to happen is either because some DNA damage happened, or perhaps some of these retrotransposons jumped around and landed somewhere else in the genome, so the genome tries to suppress that by methylating. Then you start getting a shift in the methylation pattern. So in the case of cancer, you will start getting silencing of the genes by changes in the marks, the epigenetic marks on DNA and histones. Whilst on the other hand, you start losing methylation that controls the expression of this viral retrotransposon kind of DNA. So you basically start losing control of expression with time. And because you also accumulate DNA damage with time, which also then affects the methylation patterns, you get this drift, which of course isn't a good thing. The question is, can we manipulate this nutritionally? Can we reverse the methylation changes that are happening? Okay, and this is obviously a big question in science today. We certainly know we can do it in a very crude way with agents such as 5-azacitidine, which inhibits the DNA methylase, but 5-azacitidine is the most damaging agent I've ever seen under a microscope. So I don't think it would be a very elegant, and I wouldn't call that precise medicine or precision nutrition by a long shot. Green tea, epigallocatechin gallate, also inhibits DNA methyltransferase, but exactly to what extent that is done specifically, we don't know. So we are still in early days. So generally, what you see with aging is you get an increase in global hypomethylation because most of the DNA is these kinds of elements rather than the protein coding elements. So you get hypomethylation here, and you get hypermethylation in the protein coding genes. But overall, globally, it's hypomethylation. However, if you look at specific genes, it becomes extremely precise. So Steve Horvath looked at the methylation of specific genes or promoters in specific genes. And you could see a very tight relationship between a person's age and methylation here. And this is known as the epigenetic clock, in fact. And it's thought to be probably the best way of, if there is, for example, some forensic case to try and determine the age of an individual, this is probably the most precise method available now to determine that. And furthermore, so this DNA methylation specific pattern is also associated and predicts mortality later in life. So how we can affect this kind of methylation at the protein coding genes is really, nutritionally, is an important question and one ripe for investigation. Now, the other level of control of gene expression is after transcription through microRNAs. So microRNAs are encoded from the DNA, and they are processed via proteins such as DRUSHA and dicer. So it's a long RNA that is exported out into the cytoplasm, where it is diced by this protein appropriately called dicer, and then allowing this RNA to be this RNA can then be identified by a protein here. It associates with the RISC complex, which then helps to degrade the RNA that has been transcribed, and that provides further control. So microRNAs are associated also with cancer. So I'm showing you some examples here that are upregulated by folate deficiency, retinoic acid, and vitamin D. And these are such as MIR222 is upregulated with folate deficiency, for example, and this affects the proliferation signaling that occurs in cancer. So it is thought that you might be able to control cancers by treatment with microRNAs. So what we actually see, therefore, is we've got transcription factors and microRNAs that control gene expression. And what the interpretation of the ENCODE data of these two key factors indicates that there's a very subtle hierarchical interplay in the network between transcription factors and microRNAs, which provides the fine-tuning of transcriptome and proteome regulation in cells. So the transcription factor will say, yes, let's go, we need to really express this gene, and the mRNA is saying, well, maybe not so fast. Maybe we need to pull the brakes a little bit here. Not right now. It depends on the diet or the situation, the health situation of the individual. There's a kind of, for want of a better word, a conversation that goes on before a decision is made. Now, this can, of course, be expressed as a proteome and observed as such. This is not a field I'm expert in, but there are really very beautiful and sophisticated methods to do this. And I know here at Johns Hopkins there's great capability in this field. So I'll give an example here from the work that is carried out here by a marvelous group you've got here in relation to the proteome that is associated with micronutrient status in undernourished children in Nepal. So let's say in the case of retinol, RBP4 has a very strong association with the retinol status of an individual. And the same applies for other markers for vitamin D, tocopherol, copper, and selenium. Now these markers, there will be a number of them, some of which would be better associated with nutritional status than others. So in the case of the selenoproteome, GPX1, GPX3, and the CEL-P are the ones that are really respond to nutrition better than others, and these are the ones that would normally be used. I give an example in a very crude work we do back in my lab when we look at DNA damage effects of nutrients. We use the cytome. You can see here a very strong and sensitive effect of zinc status on DNA damage markers, micronuclei, bridges, and buds. And also you can see the changes in protein expression of gamma H2AX, OGG1, which is a glycosylase that identifies oxoguanine, caspase, that is associated with apoptosis, and metallothionein, which is a known marker of zinc status. So you can do this in vitro and in vivo. Of course, the effects on proteins eventually affects metabolism and the metabolome. But the metabolome can be used in many other ways. It can also be used to understand dietary exposure. It can be used to understand the endogenous metabolome when you are not eating, probably after an overnight fast that will tell you what is happening endogenously. What happens after you eat foods? Which markers can you deduce from the metabolome that tells you what food has been eaten? And how does the medicine that you might be taking or pollutants affect this? So there's a nice study here on the metabolomics of nutritional epidemiology and identifying and markers for intake of different foods. For example, in the case of bananas, There's a negative association here with gamma tocopherol. So bananas tend to reduce the level of gamma tocopherol, and gamma tocopherol was associated with increased risk for type 2 diabetes in the very first slide I showed at the very beginning. And gamma tocopherol is also negatively correlated with the healthy eating index, as indicated here. You can use a similar approach, let's say, to understand the metabolome of B vitamins. So if you just take 3 metabolites, homocysteine, formate, and methylmalonic acid, the changes that you see can help you identify what deficiency is actually occurring. You can distinguish between vitamin B12, folate, or B6 and riboflavin deficiency, depending on how these have gone up and down. Okay, so in the case of B12 deficiency, all of these, homocysteine, formate, and methylmalonic acid, go up. But in the case, let's say, of riboflavin deficiency, only homocysteine goes up, formate goes down, and no impact on methylmalonic acid. Okay, so coming to systems approaches, well, we can therefore explore the diet at a cellular level, in vitro and culture. You can look at animal models, you can look at the human, and you can look at all the multi-omic approaches that you can use to study this. You can look at cells without even knowing the genome and seeing the impact of nutrition by simply exposing them to nutriome arrays, nutrient combinations in different concentrations, and study the impacts on the health of the cells. So even if you didn't have the money to do all the genomic stuff, you can simply start interrogating the cells of an individual in this way and find out which nutrient combination their cells will grow best in a genomically healthy way. And we use this approach in some of our studies. Another way in vivo studies is this by study done in Ireland by Lorraine Brennan's group, where they looked at the effect of weight loss using a metabolomic approach and trying to personalize it. And what they did here was they identified subgroups by principal component analysis of triacylglycerol, total cholesterol, HDL cholesterol, and glucose. And in this way identified which of these subgroups responded best to weight loss regime in this way. So they did it kind of after they did the intervention. They went back to the baseline data, classified the individuals by principal component analysis of these 4 metabolites, and showing strong differences. We're looking at p-values of minus 102 and so on, and associating it with the condition, with the phenotype of that was induced by the intervention. Another approach is one published last year, very interesting, that used the microbiome data, blood tests regarding glucose levels, dietary questionnaires, anthropometrics, what their BMI was, and food diaries, and put this into an algorithm that predicted very, very well the glycemic index response to different foods in these individuals. So this is another approach. So there's many, many ways you can use the information that is at hand. Another approach is to study an individual in all the omic ways you can think of. And here's the so-called integrated personal omics profile that was done by Michael Snyder's group on Michael Snyder himself in Stanford, which you probably know about, investigating the changes in his IPOP, so to speak, over a number of months as he changed his diet, as he was ill, and so on and so forth. And this kind of information is thought could be used to build super models or so-called patient avatars to use in precision and systems medicine. So that for each individual in the future, there may be, for you and each and every one of us, a system avatar, a mathematical model, kind of, that would predict what would happen if we changed our diet in this way and that way in health and disease. At a more ecological level, though, we have to think beyond nutrition, because our health is affected not only by nutrition, but also by our psychological well-being, our social network interaction, as well as what's happening within the body. And these effects need to be integrated in an ecosystem way to optimize health at all levels, not just at the individual level, but also at the personal network level, and even I'd say beyond the human level to the whole ecosystem and the animals that share the world with us. So this is the concept that I would like to leave you with, and I've tried to encapsulate this concept and idea through trying to put together these concepts in a manner that reflects what Hippocrates was telling us in a very clear way, except at that time these systems, knowledge and methods were not available. So I'll just read it out to you. I proposed Hippocratic Code for our pioneer doctors of genome and systems health. I swear to fulfill, to the best of my ability and judgment, this covenant. I shall endeavor to use the knowledge given to me to help my fellow beings to stay as healthy as possible at the genome, cellular, organ, system, and psychosocial level, so that they will not require to be cured from a preventable disease. I shall appropriately use the best validated diagnostics to determine with certainty the extent to which they have drifted from optimal genome and system health, and the causes of such harm. I shall use holistic methods, including nutrition, lifestyle, environmental, and socio-psychological interventions that are best suited to return my patients to optimal well-being, sustainably, and within their means. At the community level, I shall contribute to creating ecologically viable, enjoyable, stimulating environments, caring and vibrant societies, allowing equal opportunity for realizing one's purpose and creative potential while securing optimal genome and systems health to do so. This idea and thought to put these kinds of words together happened really because of a movement in Australia by some physicians who want to take this knowledge of nutritional genomics and environmental genomics and use it in their practice. So for some years they had been asking me to organize workshops on this topic to help train and educate the health professionals and doctors. So 90% of the association is known as the Australian College of Nutritional and Environmental Medicine are doctors. And that is what I was doing. And at some point we came to agree that it's time to have what's called a Nutrition and Medicine Conference, which is being now held every year. And it attracts about 400 to 500 people each year. For Australia, being a small population, is really quite a large number. And at that point in 2013, they asked me to give this kind of presentation that I'm giving you today, but this is, of course, updated a lot. And it was at that point that I started thinking, after, I think, 2 or 3 glasses of red wine, this thought came to my head. So the first version wasn't very coherent, and I'm trying to, of course, the words can be crafted a lot better than what I've put up there. So thank you very much.